Welcome back, everyone. So we're going to finish off chapter 4 of our open sex textbook, looking at the working cell. So in this last segment of the working cell, we're going to get into a little bit of physics. So we're going to learn some information about energy, as well as how it applies to us as organisms. So the biology section of it. So, but we do need a little bit of understanding of, what energy is and things like that. Okay, so we'll get into our last section, 4.3, energy and metabolism. So when we're talking about cells, of course, one of the properties of life is that living things need to acquire energy, right? And so, we have to undergo numerous chemical reactions. So all of our cells undergo chemical reactions, and metabolism is just basically the total number of chemical reactions that take place. So we're going to learn about what metabolic pathways are, and we're going to again get into a little physics and talk about the first and second laws of thermodynamics. We'll explain the difference between kinetic energy and potential energy, and we'll describe what an endergonic reaction is versus an exergonic reaction, as well as dive into enzymes. So enzymes are basically biological catalysts. Okay, so we'll learn about how they can help speed up reactions in our cells. So we're going to start off by trying to answer this question: how are energy and metabolism related? Okay, so first off, we need to understand what bioengineerics is all about. So first of all, bio means life, engineerics, think energy. So it's the concept of energy flow through living systems. So if you think about it, where does all of the energy originate from that is in your body? Well, we can trace all of that energy from the sun. Okay, so the sun is what actually powers everything on Earth, pretty much. So basically, if you think about your body is all that energy came from the food that you ate. Well, where did the food come from that you ate? Well, we can trace it back to animals and plants, right? Well, the animals that eat the plants, well, where did the plants get their energy from? They got it from the sun. Okay, so everything can be traced back originally to the sunlight. So we're going to learn about how the energy can be transferred from one form to another. So you can see here that, in our case, the energy originated from the sunlight. And then, here it's transferring into organic matter through photosynthesis into plants. And then here, the animal ate the plant, so now the energy got transferred into the flesh of the animal. And then, you either eat the animal or you eat the plant, and now it's inside of you and it's giving you energy, and you can break it down and convert it to cellular energy and so forth. Okay, and all along the way, that energy is used to power all of the chemical reactions that are occurring in your body, and some of it gets released as heat and dissipated as heat. So that's what bioengineerics is all about. It's that concept of energy flow through living systems. So you can see it, we could look at it broadly, originating from the sun and flowing through like a food chain from the plants to the animals to the decomposers. Or we can simply look at it from, the body perspective. So as it's traveling into the cells, and the cells using it to perform their chemical reactions. So the chemical reactions, what are chemical reactions? Why do we do chemical reactions? Well, if you recall from, a couple weeks ago, we talked about in chapter 2 how we build macromolecules through dehydration synthesis reaction. So that's a type of chemical reaction. So we're building those complex, big molecules from the smaller monomers through dehydration synthesis. Opposite, we can hydrolyze the reaction through hydrolysis to break apart. Okay, so that's another type of reaction. So we have these chemical reactions occurring inside of our cells all the time. And some of those reactions, they occur spontaneously, meaning that they don't require any energy for it to occur. So it can just occur spontaneously and ends up releasing energy in the process. Some of those reactions that are occurring inside of our cells actually have to have energy in order to do it. Okay, so some of those require energy. And so in order for our cells to perform all of these chemical reactions, we need to produce more energy to replenish the energy requiring reactions. So we have to basically have a constant supply of energy to power our cellular needs. Okay, so that's why, we constantly need to replenish our energy supplies by eating, 3 meals a day plus snacks or whatever. Because, we're constantly needing energy to replenish the energy that we have depleted and have been released as heat. Okay? And metabolism is basically all the chemical reactions that take place inside the cells, including those that consume and generate energy. So if you think about it, we hear that term metabolism kind of a lot in just like health related things. You want to speed up your metabolism. As you get older, you slow down your metabolism. So it kind of makes sense if you think about it. As you get older, your metabolism starts to slow down. Now, why would it slow down when you get older? Well, you're not growing anything anymore when you get older, right? When you're young, you have a fast metabolism because you're constantly growing. So you have to have all these chemical reactions occurring inside your body to allow your body to grow. But as you get older, you're not growing anymore. You're pretty much the height that you're supposed to be, and your body's starting to age. And so as it's aging, you're not having as many chemical reactions occurring in your body. So your body is starting to slow down. Your metabolism's slowing. And what happens when you start to get older? Well, think about, older people. Do they need as much food? Okay, so probably not, because they don't have as many chemical reactions occurring in their body. They're not growing. They probably aren't as active. Okay, so they don't need as much food or fuel for their body because they don't have as many chemical reactions taking place. All right, so your metabolism is simply all the chemical reactions that occur inside your cells. So if you have a fast metabolism, you need lots of fuel, lots of food to supply it. If you have a slow metabolism, you need less fuel intake, right? Less food, because you have less chemical reactions occurring. All right, so all of that's related to like our health. All right. So we're going to start out with a video. So this video, it's actually a crash course video, so I kind of switched out to crash course instead of Amoeba Sisters. And this one gives us a great overview of energy. So it talks about, the different types of reactions and so forth. It's a longer video, but it's a good introduction into this idea of energy.
Cells are like microscopic cities. They hustle and bustle with work to be done and things that need to get from point A to point B. And cities of all kinds need energy to run a whole range of important processes. We use energy to drive our cars, heat our homes, and even charge our cell phones, though mine is always just clinging to life at 5%. Like cities, cells use energy to carry out a variety of functions, all the bodily functions in fact, and all the functions of all the living organisms on Earth. But before we can understand how and why cells harness energy, we need to take a look at what exactly energy is. Sure, it's the stuff that comes out of our walls to power our homes and let John Greenblatt write his next book, The Anthropocene Rebooted. But what is it really? And what does something as seemingly abstract as energy have to do with biology? Hi, I'm Dr. Sammy, your friendly neighborhood entomologist, and this is Crash Course Biology. Who is this calling me? Right in the middle of a shoot. Sorry guys, I gotta take this. It's the theme music. Now, when I say the word energy, you might picture a lot of things. Power plants, lightning, batteries. Those are physical representations of the distinctly non-physical thing that is energy. Energy is not made of matter, but it does affect matter. We scientists define energy as the ability to cause change, specifically in the form of work. And the scientific definition of work is to move an object over a given distance. So when I say that you're over there working it on the dance floor, I mean that scientifically too. Most of the usable energy on Earth comes from the sun. But most animals like us can't use it right away. That's where plants come in. Through photosynthesis, plants combine energy from the sun with carbon dioxide and water to make sugars like glucose, which plants use as food. It's sort of like cooking with an oven. You don't eat the heat from your oven, just like plants don't eat sunlight, but they use it to transform the ingredients into food that they can eat. And when an animal eats that plant, some of the energy is transferred to the animal. So, truthishly, energy is like this swirling, whirling vibe that connects us all, bro. I'm kidding, but it is kind of like that. Because the laws of thermodynamics explain how the energy in the universe transforms and moves from one thing to another, operating as one big connected system. So how does energy work? Well, the first law of thermodynamics is that energy can't be created or destroyed. Instead, it's transformed from one form to another, or transferred from one thing to another. Like this stretched rubber band knocking over this plastic campfire. It works! Did it break? So living things exchange both energy and matter with our surroundings. So when we use our energy, whether it's to digest a burger, write a love note, or nearly break a prop, it's not available for us to use anymore. But it's not gone. It's just been transferred to something else or transformed into something that we can't use anymore to do our body's work. So when I eat that burger, I receive energy that the cow got from eating grass, and that grass got from the sun. But I still can't use that energy right away because it's chemical energy, which is a form of stored or potential energy. You can think of the chemical energy inside of plants or inside us, like a spring that's coiled up, ready to sproil. Is that what a spring does? A spring sproils? I feel like that's right. Let's fact check it though. Anyway, the point is, it has the potential to release energy, but only when it's activated in the right way. When cells need that stored potential energy, chemical reactions have to unlock it from the sugar molecules and get the work of life done. With other key reactions, this process releases potential energy to do whatever work is needed in the cell. And there's a lot of work to be done. Cells use chemical reactions to make all sorts of stuff that they need, like hormones to talk to other cells, and even to construct their own internal organelles. They also break down food and move chemicals across membranes. So it's not just the daily muscle movements that we're aware of. It's countless microscopic movements that are each essential to keep your body functioning. If a cell ever stops doing its chemical reactions, it's officially dead. That's actually the definition of dead that we use in science. The point at which an organism can't continue those chemical reactions that create order. And when a cell dies, that's it. No save points, no Phoenix down. It's game over, man. Game over. And by the way, your cells are dying all the time. In fact, you lose about the equivalent of your body weight in dead cells every year. Isn't science fun? But don't worry. Your cells get replaced all the time. It's fine. You'll be okay. Okay, the second law of thermodynamics is that disorder, or what we call entropy in the universe, is always increasing. It's sort of like how your room always seems to get messy again, no matter how often you clean it. Except in this case, the dirty clothes are atoms, and your room is the entire universe. After all, there are infinitely more ways for things to be disorganized than there are for them to be perfectly ordered. Now, chemical reactions sometimes increase the entropy or the number of ways that the molecules can be arranged in the system. This happens, for example, when bonds break so that molecules are now attracted or repelled from other molecules in new ways. And in order for life to be ordered at all, we need that chaos. You see, every chemical reaction is a balance between the changes in entropy and the changes in the energy that happen during that reaction. To find out why, let's head over to the thought bubble. It's time for a crash course camping trip. In our campfire, wood and oxygen are our reactants, or the raw materials for our reaction. Once the fire gets going, the wood and oxygen undergo a chemical reaction to form new products, or the results of the reaction. In this case, CO2, water, and other compounds. The energy contained in the bonds of the original reactant is more than the energy contained in the products. In other words, energy that was stored in the orderly bonds between the molecules in the wood has been released. Larger molecules are broken into smaller molecules, ripe for new combinations, and entropy is increased. That makes this an exergonic or energy-releasing reaction. Energy-releasing reactions are considered spontaneous, but that doesn't mean that they'll start on their own. Spontaneous just means that once exergonic reactions start, they don't need outside energy to keep them going. On the other hand, there are reactions that are not spontaneous. They require work. Take, for example, the photosynthesis that occurred in the tree before it became part of our fire. In this endergonic reaction, energy is essentially absorbed rather than released. The tree took molecules of carbon dioxide and water bouncing around in the air and soil and whipped them into shape, a very specific woody shape. But whether it's a tree converting sunlight into energy, or you dropping that tree's hard-grown wood into a fire pit, every reaction needs activation energy, something to set it in motion initially, like the spark of a match. Thanks, Thought Bubble. So while all exergonic reactions do happen spontaneously, sometimes the required activation energy to get them started is such a lift that the actual reactions happen very slowly. Too slow, in fact, to keep our bodies alive and functioning if they happened on their own time. Thankfully, nature has a trick to speed up these reactions. Proteins called enzymes. Enzymes stick to reactants because they have a complementary shape, like puzzle pieces. Then they catalyze reactions, speeding them up by doing things like pushing two reactants super close together, or putting stress on a bond that needs to break. By getting the chemicals closer to their reactive form, the enzyme lowers the energy it takes to activate the reaction, making it happen much faster. And the cool thing about these enzymes is they can be used over and over. The reaction that occurs doesn't permanently alter the enzyme, so it just keeps going and going. Enzymes power reactions in more life functions than you can count, both in and outside of the human body. One cool example, newborns need to eat a lot. So there's a special enzyme in breast milk that helps babies absorb fat more quickly. Or sometimes, enzymes help reactions team up in what's called coupling. See, when two reactions love each other very much and they want to spend their lives together, not that kind of coupling. When enzymes facilitate coupling, it allows the energy generated from an exergonic reaction to power an endergonic reaction, sort of like a stationary bike that couples the kinetic energy of pedaling with the electrical energy to charge your phone. Most living things on Earth rely on enzyme-facilitated coupling to break down the carbohydrate glucose and use it as a major source of energy. And that's just to name one example of many. Usually, we need more than one reaction to occur to build a complex molecule or to break it down. When a series of enzymes helps lower the activation costs of a series of chemical reactions in this way, we call it a metabolic pathway. A metabolic pathway either breaks down or builds some specific biological molecule that the living thing needs for a particular function. For example, these pathways help assemble complex molecules like nucleotides, which are the building blocks of our DNA. And the more we understand about how these processes come together, the better we'll get at fighting a number of diseases. Cancer and diabetes, for example, are often treated in part with enzyme inhibitors, which work to slow or block the enzymes that aren't working as they should. And amazingly, all living things have strikingly similar metabolic pathways. So when we learn about ourselves, we also learn about other species. And likewise, when we study other organisms, we learn more about ourselves and the energy that we all share in this big, connected universe, man. Just like energy powers work all throughout a city, energy in our cells powers cellular work. which keeps our bodies running in so many beautiful ways. Whether you're playing video games, doing homework, or mourning your billions and billions of dead cells, your body is using energy. Energy that's involved in chemical reactions happening within you every moment of every day, with a little help from enzymes. In our next episode, we're going to take what we learned about ATP and metabolic pathways a few steps further to talk about cellular respiration. I'll see you then. Peace! This series was produced in collaboration with HHMI by...
Okay, so again, that was a great introductory video into this whole idea of energy, and like Dr. Sammy was talking about, it's even going to lead us into next week when we talk about cellular respiration. So it gives us a good foundation into what we're studying in this chapter as well as next chapter. So let's move on to this idea of metabolic pathways, which he talked about in the video. So metabolic pathways, we talked about metabolism being those chemical reactions that occur inside your cells, so that all of those chemical reactions. So metabolic pathways are just a series of reactions that take place to yield a final product. So sometimes, we have to go through a series of steps to get that final product. Just like you can think of when you are, making some sort of, toy or something like that, where you have a series of steps that you have to take until you end up with that final end product, that finished toy. So a metabolic pathway is the same thing. It's just a series of reactions that have to take place, starting with one molecule and then undergoing another reaction to modify it to another molecule, and then undergoing another reaction to modify it to another molecule, until eventually you'll yield the final product. So you end up with like a series of these intermediates until you end up with that final product. Okay, and each time you have a reaction take place, there's an enzyme that's catalyzing that reaction. So like the video talked about, reactions, they don't occur necessarily spontaneously. I mean, yeah, some of them don't require much energy, but you have to get past that activation energy in order for the reaction to take place. So we have to catalyze those enzymes. So every single reaction has to have a separate enzyme. Okay. So to start us out with some of those chemical reactions that occur in our body, we're going to describe two types of reactions. We have what are called anabolic reactions and catabolic reactions. So anabolic reactions are when we have small molecules built into larger ones. Okay, so in this case, we require energy to combine those molecules together. So we're taking these small molecules and we're building something large. So energy is required in order for that to take place. Now, this kind of sounds like another type of reaction. It kind of sounds like dehydration synthesis reactions. Remember when dehydration synthesis reactions, where we take our monomers, we remove water in order to bring the monomers together to form the polymers. So that would be an example of an anabolic reaction. It requires energy in order to bring those two monomers together. Okay, so that would be an anabolic reaction. So another example would be, like from the video, photosynthesis. Taking carbon dioxide and water, you can't just expect them to just spontaneously come together and form glucose. Okay, you need to add energy. You need light energy from the sun in order to do that. So that is an anabolic reaction. Okay, so anabolic reactions where energy is required to bring small molecules are built into large ones. Now the opposite is a catabolic reaction. So catabolic reactions are when we have large molecules broken down into smaller ones. And in this case, energy is released. Okay, so breaking those bonds between the molecules releases energy. So a hydrolysis type reaction, which we learned about earlier in the term, that would be an example of a catabolic reaction, because breaking those bonds by adding water, it releases energy. Okay, so energy would be released. Another example of a catabolic reaction, which we'll talk about next week, is cellular respiration. So by taking that glucose molecule and undergoing cellular respiration, we will produce carbon dioxide and water in a burst of energy, so ATP. All right, so energy gets released from a catabolic reaction. Now, one way that I kind of remember the difference between the two is catabolic. I think cats are full of energy and they break things. So I think, cats, they're running around, breaking things, releasing a bunch of energy in the process. So catabolic, think cats running around, breaking things, releasing energy. So that's, how I remember catabolic versus anabolic. So maybe that'll help you remember the difference between the two. All right. Moving on, energy. So what is energy, first of all? So energy is the ability to do work. So the study of thermodynamics is basically where we're learning about or studying energy and how energy is transferred, okay? So that involves like physical matter. So basically looking at how energy is transferred from the plant into the cow, and then how that becomes the hamburger that fuels the body of the person, like we saw in the video. And so when we're thinking about energy, we think of it as basically a system is the matter relevant to a particular case of energy transfer. So matter, you could think of it as anything, like a living person would be a system. And then how energy is transferred into or out of that system. So the surroundings is anything outside of the system. So if we're talking about an open system, energy can be exchanged with its surroundings. If we're talking about a closed system, energy cannot be exchanged with its surroundings. So in biology, biological organisms are open systems because one of the properties of life is that energy has to be transferred, or we have to acquire energy from our surroundings. And so we think of ourselves as an open system. We need to acquire our energy. So us as humans, as consumers, we eat our energy, right? Plants, where do they get their energy from? They're an open system too. They acquire their energy from their surroundings as well. They take light energy from the sun and they convert it to chemical energy using photosynthesis. So they are also an open system. Okay, so in biology, our energy systems are always open. Okay, so we're not talking about closed systems in this field. Okay, so that would be something else. Now, from the video, he talked about the two laws of thermodynamics, so we'll go through those. So with energy, the first law of thermodynamics states that energy cannot be created nor destroyed. Okay, so it can only be transformed or transferred into different forms. So a power plant is not making energy at all. Okay, so that's a big scheme. Okay, so a nuclear power plant is not creating any energy. So that is, big coal power plants, they don't make any energy. What they do is they transfer energy from one form to another. So they, take, for example, a coal power plant. So what does a coal power plant do? It takes coal, which is basically a bunch of carbon, and burns it, right? And when you burn coal, you can heat up water or whatever, and so that steam can cause turbines to move, and then you can kind of convert that into electricity. So you can convert, an unusable form of energy into a usable form of energy. So the first law of thermodynamics is not not in conflict there by a coal power plant. It's not creating or destroying energy. It's just transferring it from one form to another. It's using the energy stored in coal, but just transforming it into a usable form, electricity. Okay? And we can't destroy energy. So energy never gets destroyed. So even when you eat something and your body uses that energy, it never goes away, or it doesn't get destroyed. It just gets transferred into something else. It gets transferred into heat, which dissipates into, when you exercise, when you use up your energy, that dissipates as heat. Okay? Or, it leaves your body as waste. And then, that's the energy has left your body as like solid waste or as heat. Okay? So first law of thermodynamics states that energy cannot be created nor destroyed. It's only transformed into different forms. Okay, so here's an example. We can have our chemical energy from our food. We can transform it into, being able to move, okay, with it, kinetic energy. Here's light energy from the sun, can be transformed into chemicals through photosynthesis. The second law of thermodynamics states that energy will always be lost as heat in energy transfers or transformation, and that increases entropy. Okay? So every time you have a chemical reaction occur or an energy transfer occur, it's not going to be a 100% transfer. So if I take, 1,000 watts of energy from coal and transfer it into electricity, I'm not going to get 1,000 watts of electricity from that. Okay, so that it's not going to be 100% conversion. Some of it's going to be lost along the way. Now, it's not going to be destroyed. It's just going to be lost, and that loss will always be heat. It gets dissipated as heat because of this idea of entropy. Entropy is like chaos or disorder. So it's that idea that it's not going to be 100% energy efficient. So if you think about, like, for example, when you eat your food, let's say you have 2,000 calories a day. Does that 2,000 calories go straight to your body and add 2,000 calories to your muscles? Do you add, a pound of flesh every time you eat every single day? No, because then you'd be like 3,000 pounds, okay? Because a lot of it gets lost as heat, right? You have chemical reactions that are constantly occurring, so everything gets, it's not a 100% transfer of energy from flesh to flesh, from eating the hamburger to, adding that protein onto your bones. Okay, some of it gets lost along the way as heat. It's not 100% transfer. Okay, so that's due to that second law of thermodynamics. Okay, so some of it will get lost. All right, so now that we know about how energy is not created nor destroyed, only transferred, now we can apply it to potential and kinetic energy. Okay, so let's take a look at this example of, we have a dam here with water behind it. So behind this dam, this water has a lot of energy, but that energy is not in motion yet. Okay, so this is what we call potential energy. It's stored energy because it's behind this dam. But the moment we release the water from this dam, that water starts to move. And now that energy is in motion. So now it becomes kinetic energy. So potential energy, think of stored energy. Kinetic energy is energy in motion. So when we are about to bite into an apple, okay, that apple is potential energy. Okay, so I mean, we haven't eaten it yet, we haven't, used that energy in any way yet, but that's just, a bunch of energy sitting there in that apple. So that's potential energy. But once we, chew that apple and digest the nutrients and bring it into our body and then start burning the calories and moving, that would be kinetic energy. So potential energy is stored energy. Kinetic energy is energy in motion. Okay. Another example would be, for example, like let's say that you are on a diving board and you're about to dive into the water. So before you leave the diving board, you're at potential energy. Okay, so you haven't left the diving board yet. So you have the potential to leave the diving board. So you are at the maximum energy level. But the moment that you leave the board, your muscles start moving, the air is starting to flow around your body, then the energy moves to kinetic energy associated with motion. Okay, so there's the difference between the two. Stored energy versus energy associated with objects in motion. So water flowing down the stream, that's kinetic energy. Water behind the dam, potential energy. All right. So how do we apply this to our chemical reactions? Well, when we have our chemical bonds holding the two atoms together, that's potential energy. Okay, so potential energy exists within the chemical bonds. And when the chemical bonds are broken, then that energy is released. And we call that chemical energy. Okay, so chemical energy is, the energy that gets released when those bonds are broken. Okay, so if you think about a polysaccharide that we learned about earlier, like starch, that's a bunch of potential energy because there's a bunch of chemical bonds holding each of those glucose molecules together in that starch molecule. So that is potential energy. And then when we break apart the bonds, the energy can then be released as chemical energy and be used for processes in the body. All right? So that's where we get our chemical energy from. Okay, so now that we have an idea, we've gone through a lot of terms here. We've talked about anabolic reactions, okay, that require energy, catabolic reactions that release energy. We've talked about we just talked about first law of thermodynamics, states that energy cannot be created nor destroyed, only transferred. Second law states that every time you transfer from one form to another, energy is lost as heat. We talked about kinetic and potential energy. Now we're going to get into something called free energy. Okay, so free energy is energy that's associated with a chemical reaction. All right, so if we have in a chemical reaction, if we have negative amount of free energy, that indicates that energy was released during the reaction. Okay, if we have a positive amount of free energy, that indicates that energy was gained during the reaction. And by the way, instead of writing free energy, we use this symbol, it's delta G. Okay, so if you see delta G, if you see negative delta G, that indicates that energy was released. If you see positive delta G, that means that energy was gained during the reaction. So this can help us determine if the reaction is spontaneous or not. Okay, so if energy was released, that means it was a spontaneous reaction. So that means it's what we call an exergonic reaction. So an exergonic reaction is a spontaneous reaction. So that's where energy is released, resulting in that negative free energy. So let's take a look at this diagram over here. So here we have the energy level on the y-axis, and then the reaction time on the x-axis. So we have the reactants, so that's, you know, if we have a chemical formula or chemical equation, that's on the left side. That's, you know, like for example, if we're doing, I don't know, cellular respiration, that would be glucose over here would be a reactant, and then the products are the, you know, what results, the carbon dioxide and water. So in this case, we have our reactants. So they start out here. We need a little bit of energy to get it started. So every single reaction, we do have to have some energy to get the reaction started. And that's called activation energy. And we'll talk about that in just a minute. Okay, so that's just a little bit of energy to get the thing started. Okay, and all reactions have to have that. So once that occurs, it gets it started. Now in this case, we have so much energy to begin with. Look at how much energy we have to begin with, that by the end, the products, the end result has less energy than what we started with. Okay, so we have less energy than we started with. That means that energy was released during that process. So that's an exergonic reaction. So energy got released. So an example of that type of reaction from the video we saw was like a fire burning, so like a campfire. So if you have logs are basically just a bunch of carbon, right? You have, you know, trees, they are basically just a bunch of carbon. And you ignite them, and basically it's just burning a bunch of carbon. And once that happens, you know, you don't have to keep adding energy to get the fire keep the fire going, right? It just, you know, the logs just keep burning and burning and burning until there's no more fuel left. And so you have lots of energy in that log, right, to burn it. So energy is released in the form of heat from that fire, okay, until you end up with just ash at the end, okay? So that ash is just the end product. So think of this reaction as burning a fire, burning logs, okay? So the logs had a lot of energy to begin with. You're burning it, lots of fire, heat is released, you end up with ash at the end. You can't burn the ash, right? There's no more energy left. It's basically just ash. All right? So that's an exergonic reaction. Okay? The opposite of that is an endergonic reaction. So endergonic reaction means that we don't have hardly any energy to begin with. So we have to add energy to get the reaction to take place. And then we end up with the products that have more energy than what we started with. Okay? So this is not a spontaneous reaction. Okay? This requires energy to take place. And the video talked about photosynthesis being an example of an endergonic reaction. So the reactants in photosynthesis are carbon dioxide and water. Now, there's carbon dioxide in the atmosphere, and there's water in the atmosphere. And there's no way that you're going to get sugar just spontaneously being made in the atmosphere, right? Okay, because there's no energy there. All right, so inside the plant, you have the chloroplast. And inside the cell of the plant has a chloroplast. And so the plant takes that, you know, carbon dioxide and the water inside the chloroplast, and then it requires energy from the sun. That sunlight is the energy that's going to be used to smash the water and the carbon dioxide together to form glucose. Okay, so now you have a product, glucose, that has more energy than the reactants, carbon dioxide and water, had to begin with. Okay, so you ended up with positive free energy. All right, so that's endergonic, requires the addition of energy. All right, so think exergonic, you release energy like burning a fire. Endergonic, you have to add energy to produce something, so photosynthesis, add sunlight to make the sugar. Okay. So let's see how well you understood the concept of endergonic versus exergonic. So we have 4 pictures here, and we're going to walk through each one of these pictures and try to figure out if the picture is showing a process that is endergonic or exergonic. So remember, endergonic, you have to, requires the addition of energy in order to form the product. Exergonic, energy gets released. So let's start with this right here. So this is a compost pile. So the person, adds basically a bunch of wood chips and, leaf litter and stuff like that. And then over time, it breaks down. And during that process, it's breaking down and turning into soil. So the compost pile is decomposing. So is this an example of an endergonic or an exergonic process? So a decomposing compost pile. If you chose exergonic, I would agree with you. So in this process, exergonic, actually, if you've ever had like a compost pile and it's in the middle of winter time, you might actually see it steaming, okay? Because it's releasing energy. So basically, you can actually feel warmth being produced in there, like heat is being released. As those microbes are breaking down all the organic matter and breaking it down into smaller substances, and during that process heat is released, so energy is being released in this process to break down the wood and all sorts of materials into smaller components. So this is an example of an exergonic process. Okay. How about, here's one, we have an example of a little chick. Okay, so how about like a baby developing from a fertilized egg? Is that an example of an endergonic or exergonic process? A baby developing inside an egg, fertilized egg, or from a fertilized egg. Well, we're building something, right? So in that case, since you're building, you're starting out with 1 cell, and the cell divides into 2 cells, into 4 cells, and 8 cells, and then it becomes this little complex little baby chick. That's a building process. It requires energy, right? So that's an endergonic process. Okay. How about this? This is where we have, basically here's a glass of water and a tea bag is placed in there. And we're dissolving tea in the water. Is that an example of an endergonic or an exergonic process? So just dissolving tea in water. Does it take any energy for the tea to spread out in the water? No, that's just simple diffusion, right? So that's just an exergonic process. So that's just requires, it's just a spontaneous reaction. And then how about this one where we have a ball rolling down a hill? Is that an example of an endergonic or exergonic reaction? Well, that's like potential energy moving into kinetic energy, right? So that is energy of motion, and that is spontaneous. It does not require, any addition of energy. So that's exergonic. Energy is released. All right. Okay. So we were talking about earlier about how in order for a reaction to take place, it doesn't matter if it's exergonic or endergonic, we do need to have a little bit of activation energy for the reaction to take place. So we're going to watch a brief video. This is actually a video found in your textbook about activation energy.
Where does the energy for chemical reactions come from? Many chemical reactions, even exergonic reactions, require an input of energy to get started. On ancient Earth, before the formation of macromolecules, chemical reactions were powered by lightning, sunlight, and heat from molten rock. On the modern Earth, living organisms, including humans, most often obtain the energy they need from other molecules. Cells use the energy in these food molecules to power chemical reactions. However, the energy cannot be used directly. It must be transferred first to an energy-carrying intermediary, such as the compound adenosine triphosphate, or ATP. When chemical bonds in ATP are broken, energy is released. This process provides the energy needed for chemical reactions which power life. This graph shows a hypothetical reaction between atom A and a molecule containing atoms B and C. The activation energy, EA, is the amount of energy needed to get the reactants to the transition state in which bonds are broken and new bonds are formed. This transition state usually lasts for a fraction of a second. In this example, the products of the reaction are the molecule AB and the atom C. Because this is an exergonic reaction, some of the energy stored in the reacting molecules is released. Often, activation energy can be a barrier that prevents a chemical reaction from occurring, except very slowly. The activation energy for many chemical reactions can be reduced with catalysts. Without catalysts, many of the chemical reactions of metabolism would occur too slowly to support life. Catalysts stabilize the reactants in the transition state, allowing old bonds to break and new bonds to form. Even though the activation energy is lowered by a catalyst, the amount of energy released by the reaction is unchanged. In living organisms, most chemical reactions are catalyzed by proteins called enzymes. The three-dimensional structure of an enzyme is crucial to its function. If this structure is disrupted by exposure to high temperatures, for example, the enzyme stops working. The enzyme binds reactants in its active site, which is specially shaped to fit the reactants. The reactants on which enzymes act are called substrates. In this example, glucose is the substrate of the enzyme hexokinase. The enzyme and substrates are represented here in an artist's drawing. The substrates include the atom A and the molecule BC. The substrates enter the active site of the enzyme. By holding these atoms close together, the enzyme reduces the amount of energy the atoms need to enter the transition state. In the transition state, the bond between atoms B and C is broken, and a new bond between A and B is created. When the new bonds form, the products are released, and the enzyme molecule is ready to catalyze another reaction. Each enzyme is specific for only one reaction. For example, this enzyme could not catalyze a reaction between atom A and molecule BD. The fit between an enzyme and its substrate is described by the lock and key model. The enzyme is the lock, and the substrate is the key. Only a specific substrate will fit the enzyme's active site. Another model of enzyme-substrate interaction is called the induced fit model. It proposes that once the enzyme binds the substrate to the active site, the enzyme changes shape slightly to bind the substrate even more firmly. This places a strain on the existing bonds in the substrate, thereby lowering the activation energy needed to break the old bonds and generate new ones.
All right, I'm not quite sure why there is like sounds in the background of that, and then there was that next video like popped up in the middle. But anyway, that was a good introduction into like activation energy. So also into what enzymes are. So, we'll talk more about that. So what are enzymes? So enzymes, they're basically biological catalysts. So they help catalyze or speed up chemical reactions. And they do that by lowering the activation energy of the chemical reaction inside the cell. So here again is an example. And first off, what type of reaction is this? Is this an exergonic or endergonic reaction? So you can see here we have less energy than we start with less energy, we end with more energy. So that would be an example of an endergonic reaction because we have added energy. So positive free energy there. So this is endergonic. But whether or not it's exergonic or endergonic, we still have activation energy that's required to get this this reaction to take place. And so what activation energy, that's that little amount of energy that is needed for the reaction to take place, and enzymes, they can lower that. Okay, so you can see here without the enzyme, the activation energy is greater than if an enzyme is present. So the enzyme actually lowers the amount. Okay. And what's unique about enzymes is that enzymes can do this over and over again because they're not going to be changed at all in this process. So once they are, part of this reaction, the products are released, they can pick up new reactants and do it again, over and over again. So they're not, they're not going to be used up in a reaction. And we'll talk more about that in a second. So we're going to watch another video that came from your textbook. So hopefully it has better background. No kids in the background this time, hopefully.
Most of the chemical reactions that occur in your cells do not occur spontaneously. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. In fact, if it weren't for enzymes, chemical reactions would proceed too slowly to support life. This graph illustrates how enzymes speed up chemical reactions. Remember that in a chemical reaction, the reactants interact to form a product. The chemical reactants for this example are shown on the left, and the products on the right. The wall that separates them represents the activation energy. You can think of this wall as an energy speed bump. The larger the bump, the slower the reaction. The yellow speed bump represents a chemical reaction without an enzyme, and the orange speed bump represents the same reaction with an enzyme. As you can see, the orange speed bump is a lot lower. This is because the enzyme acts to physically bring the reactants together. By doing so, it increases the efficiency of the reaction and lowers the amount of energy needed for the reaction to occur. Since less energy is required, the reaction occurs at a faster rate. Notice that the enzyme does not influence the energy level of the reactants or the products, but only the amount of energy that is required during the process of the chemical reaction. Without the use of enzymes, many of our body's processes, such as digestion and the processing of nerve impulses, would simply occur too slowly. The addition of enzymes, however, speeds up these chemical reactions, making life as we know it possible.
All right. So again, with the enzymes, they don't change the amount of free energy, okay? So they're only dealing with that activation energy. And so let's learn about how enzymes actually do this, okay? So here is an example of an enzyme. Remember, enzymes are mostly proteins, okay? So they're protein-based. And here we have an enzyme, and you can see that enzymes, they have like a special little area where the reactants bind to it. And we call the reactants the substrate, by the way. So that substrate could just be one reactant, or it could be multiple reactants, depending on, whatever is on the side of the equation. So here we have a substrate. So that substrate is going to bind to the enzyme at what we call the active site. Okay, so that active site is, that's going to be where the substrate binds to. And that active site is specific to the substrate. Okay, so only that particular substrate can bind to it, not some other molecule. So enzymes are very specific to the substrate. You can't have, one enzyme binding to a different substrate. Okay, so for example, if you, we talked about in last week, was it last week? Yeah. With our macromolecules, or actually the week before last, with our macromolecules, we talked about lactose intolerance, lactose being a disaccharide, so a dairy sugar. And lactase is an enzyme that breaks down lactose. So that enzyme lactase will only break down lactose. Okay, if I put maltose in with lactase, lactase won't do anything. Okay, maltose can't bind to lactase. So lactase is an enzyme that its active site is designed only to bind to lactose. Okay, so the enzyme active site is specific to the substrate. Okay, so once the substrate has bound to it, there's a special, because you have, this is a protein, and remember with our back again with chapter 1, we talked about the macromolecules, and you have the, actually, was it chapter 1 or 2? I can't remember. With the folding of the protein, you have the primary structure, secondary structure, tertiary structure. Remember with the primary structure, you just have the amino acids, and then you start the folding with the secondary structure that involve the hydrogen parts forming hydrogen bonds, and then the tertiary structure formed with the R chains. So this is where the R chains can get involved with the substrates, interacting with the substrates to form what's called an induced fit. So the enzyme will actually shift a little to form an induced fit where it almost kind of hugs that substrate. And as it's doing that, it's bringing that substrate even closer into the enzyme. And that allows that reaction to proceed. And so now we have the reaction proceeding, we end up with the products. Okay, so here's the products. Now the products will get released. Now that whole process, we have the products being released. The enzyme's still there. The enzyme hasn't been used up in any way. Now it can, go back and receive another substrate. Okay, so it can go back to normal, it receive another substrate, do the whole thing over and over and over and over again. It never gets used up. So enzymes, they can, proceed to do the same reaction over and over again as long as there's a substrate to react with. All right, so that's how enzymes work. So we're going to watch an Amoeba Sisters video on how they can, basically enzyme activity. So how do enzymes, what can regulate them? So what can inhibit them, what can cause them to work better, and so forth. So we'll watch that next.
Have you ever noticed how, when learning more about a science topic, we can get so interested that we start to discover favorites? Favorite dinosaur? I've found quite a few opinions there. Favorite animal? I'm a little obsessed. Favorite flower? Well, sometimes a little too obvious. Of course, you can always get a little more unique. Favorite protist? Favorite amino acid? Your favorite nitrogenous base? I'm a little sad that no one has yet asked me those 3 above, nor have they asked me what I've been waiting to be asked, and yet have often volunteered. What is your favorite enzyme? ATP synthase is mine, but it's a hard choice because there are a ton of fascinating enzymes. We already have an intro video about enzymes, but this video topic is about to go into a little more. Enzyme examples, cofactors and coenzymes, inhibitors, and feedback inhibition. Enzyme examples are important because without learning about some real-life examples, it's kind of hard to connect to how important they are to living things. Let's start with some fascinating enzyme examples in the human body. A great place to focus on digestion. Many enzymes focus on breaking down certain biomolecules. You remember the 4 biomolecules: carbohydrates, lipids, proteins, nucleic acids. These biomolecules are found in your food. You've got to break those biomolecules down in digestion. Amylase is an example of an enzyme that breaks down carbohydrates, and one action location is in the mouth. Amylase can break the glycosidic linkages in starch, which is a large carbohydrate, into smaller carbohydrates. Lipase is an example of an enzyme that breaks down lipids, and one action location is in the small intestine. Lipase can break the ester bonds found in triglycerides, which are a type of lipid, into their building blocks: fatty acids and glycerol. Pepsin and trypsin are some examples of enzymes that break down proteins. An action location for pepsin is in the stomach, and an action location of trypsin is in the small intestine. These enzymes break the peptide bonds in proteins. Pepsin breaks down proteins into peptides. You even have nucleases that break down nucleic acids. That's DNA and RNA. And yes, DNA and RNA can be found in your food. Nucleases break down phosphodiester bonds found in connected DNA and RNA and helps break them into nucleotides. By the way, once all of these biomolecules are broken down into smaller pieces, there are even more digestive enzymes that can break them down even more. But it's not just digestion. You can name any human body system: excretory system, respiratory system, circulatory system, and you'll find there are enzymes involved. These enzymes might break down or build up the substances that they act upon, which is needed by different body systems. And while you're at it, realize that enzymes are not just for humans. Enzymes are found in all living organisms, like this Venus flytrap here. Beyond the 3 domains of living organisms, you'll even find some types of enzymes in viruses. Okay, so moving on. What's next on our list? Oh yes, cofactors and coenzymes. So just a quick review from our main enzyme video. This is an enzyme here, and we have our active site and a substrate that could fit in the active site for the enzyme to act upon. It's an enzyme-substrate complex when the substrate is bonded in this active site. And there is likely induced fit, where the active site actually changes its shape slightly for an ideal fit, an enzyme-substrate hug. But the thing is, enzymes often don't do it alone, and they tend to have cofactors, which tend to be inorganic, like zinc or iron, or coenzymes, which tend to be organic, like many vitamins. Cofactors and coenzymes may be permanent or temporary, which often depends on how they're bonded. A cofactor or coenzyme may sit in this active site here. It helps the bonding of the enzyme and substrate so that it can work at its best, and sometimes it won't even work at all without them. An example of a cofactor or coenzyme? You know when we've mentioned DNA polymerase, an enzyme in DNA replication? It often has a zinc ion as a cofactor to help. There are also inhibitors. They might be reversible or not reversible. A lot of that depends on how they bond. They may be competitive or non-competitive. Competitive inhibitors tend to sit right here in the active site, blocking a substrate from being able to bind. The substrates are competing with the competitive inhibitor for binding. Non-competitive inhibitors bind to some other area of the enzyme, somewhere that isn't the active site. This can be referred to as an allosteric site, a site on the enzyme that isn't the active site. When the non-competitive inhibitor binds on the allosteric site, the enzyme has a shape change. If the substrate still binds, the enzyme's active site might not function well, or at all. You know, inhibitors might sound like a bad thing, but inhibitors can be harmful or they can be helpful. Some examples. You might remember DDT from our biomagnification video. It can act as an inhibitor for certain enzymes in the human body and can cause health issues. But understand that our body also uses inhibitors all the time to control a lot of biological processes. It's called feedback inhibition. This is a hypothetical example here, but let's say that enzyme 1 converts substrate A to intermediate B. Then enzyme 2 converts B to C. Then enzyme 3 converts C to a final product D. These enzymes are all working in a pathway here. But what if you have enough product D now? It's the ultimate product, but you have enough, and it's wasteful to keep producing it, or maybe it's even harmful to make too much. Well, product D might also be a non-competitive inhibitor for enzyme 1, stopping enzyme 1 from working, which will stop the process. Again, this could be reversible, or it could be that more enzyme 1s are produced in the body if this needs to start up again. So to recap, we explored some real-life enzyme examples, talked about cofactors and coenzymes, inhibitors, and feedback inhibition. But one last thing. We're big on asking why. What is the significance of learning this? So we know that enzymes are ubiquitous, love that word, and they are critical for understanding biological processes. They're also important to understand because many of the medications we use to treat diseases and disorders act on enzymes. One example, high blood pressure. It can be harmful to your organs and blood vessels. One class of blood pressure medications is the class of ACE inhibitors. ACE inhibitors work by blocking angiotensin-converting enzymes. When inhibited, these enzymes can't convert angiotensin to angiotensin 2. Angiotensin 2 can be involved with raising blood pressure, so by blocking the enzyme that helps produce it, it can lower blood pressure. There's some more complexity with this to explore. By the way, it's not just the human body. Medications can act on enzymes and microbes that make us sick. For example, penicillin. Penicillin is an antibiotic, but it works by inhibiting the enzyme transpeptidase, which prevents many types of bacteria from being able to build their cell wall. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So from that video, we learned quite a bit about what can affect enzyme activity. So we, talked about cofactors and coenzymes and things like that. So we're going to start out by learning a little bit more about how enzymes work. Okay, so enzymes, they, because they're proteins, they can be affected by changes in the environment. So enzymes, they don't tend to work optimally if in certain environmental conditions. And so they can, in fact, we talked a bit about this earlier when we were talking about proteins, they can denature fairly easily if they go outside of their optimal range. So, they do have a specific optimal range for their pH, for example, or their temperature or their salt concentration. So for example, in your stomach, like the video talked about, you have pepsin, which is an enzyme that helps break down protein. And that particular enzyme, its ideal pH is around pH 2. So, it works well in that pH, but the moment it enters your small intestine, the small intestine's pH is about a pH of 7 to 8, and so it will denature pretty easily, which is fine because you don't want pepsin in that area of your body anyway. Your enzyme activity is controlled by a number of factors. And here you can see kind of, what happens with temperature. So as the temperature increases, that could be a good thing for, a while. So as the temperature increases, this could speed up the reaction rate for that particular enzyme. But eventually it could get too warm. So if the temperature gets too hot, it can cause the enzyme to denature. Okay, so that can affect the activity of that enzyme. So think about what happens if, why our body is always about 97 degrees Fahrenheit or about 37 degrees Celsius. It's because that's where our enzymes work optimally. If our temperature gets too high, let's say around 105 degrees Fahrenheit, it can actually cause some of our enzymes to begin to unravel, and that can cause a number of issues. It could cause us to go into shock and things like that. Our body can start to shut down after that. And so, enzymes, again, they can be controlled by those environmental factors. Also, like the video talked about, some enzymes, they can actually work better with the help of cofactors and coenzymes. So cofactors, those are inorganic ions, whereas coenzymes are organic. So an inorganic ion, well, think about, first of all, a multivitamin. So if you take a multivitamin, you got your little minute quantities of several things. So you have your vitamins and minerals in there. So some of the minerals in there might be iron, might be magnesium, you might have zinc, things like that. So that might help with your cofactors. You also have vitamins in there, right? You have like vitamin A, vitamin C, things like that. Those are coenzymes, organic helpers. So the cofactors, an example of a cofactor, let's see, something like zinc. In order for an enzyme called DNA polymerase to work, which is something that helps build DNA molecules, it actually requires a little bit of zinc, that ion, to help. Coenzymes, they can be used as well. So that would be, again, some of our common vitamins, like vitamin C is a coenzyme for a lot of different enzymes to help build things like collagen and things like that. So there's a number of different coenzymes and cofactors that are used to help enzymes do their thing. Okay. The video also talked about how we can decrease enzyme activity through competitive inhibition and non-competitive inhibition. So in competitive inhibition, we can have a molecule actually bind to the active site. So if we have a molecule binding to the active site, then the substrate that's supposed to bind to it can't bind to it. And so we don't have a reaction occurring if the substrate can't bind to it, right? So that effectively stops the enzyme from functioning. If something's binding to its active site, then the enzyme can't actually do its job. And so competitive inhibition, you have some molecule competing with the substrate. Okay, so it's binding to the active site, preventing the substrate from binding to it. And that prevents the substrate from, having the reaction take place. In non-competitive inhibition, it's, similar to competitive. It's just instead of the molecule binding to the active site, it's binding to another part of the enzyme. And in doing so, it changes the shape of the active site. Okay, so it prevents the substrate to bind to the active site because now the active site is differently shaped. It's misshapen. Okay, so where that molecule has bound to the enzyme, that new site is called an allosteric site. Okay, so that allosteric site is kind of bumps into the enzyme in a sense that now that it's changed the active site, preventing the substrate from binding to it. So it's non-competitive because it's not actually sitting on the active site, but it's changed the site enough that the substrate can't bind to it anymore. Okay. So this is just kind of showing, like normal enzymes, it's going to do its reaction normally. Competitive inhibitors, it's going to, affect the rate of reaction. Non-competitive inhibitors affect the rate of the reaction. Okay. We talked about allosteric inhibition. So that's, again, here we have our enzyme. Here's the active site where the substrate is supposed to bind to. So if we have a non-competitive inhibition, that means that we have the molecule, the inhibitor, is binding to another area outside of the active site on the enzyme. So that new site is called the allosteric site. And when it does so, it changes the shape of the active site. So now that active site is no longer a shape that the substrate can bind to. So that effectively prevents the substrate to bind to it, and so that prevents the enzyme from working properly. So you get non-competitive inhibition. Okay. Now you can also have the opposite occur. You can have allosteric activation, where you can actually have the activator attached to the enzyme at an allosteric site that can change the shape of the enzyme to reform the enzyme to the correct active site so that the substrate can then bind to it. Okay, so it's basically kind of, making the enzyme work again. Okay, so you can kind of control the enzyme's activity through allosteric inhibition and allosteric activation. So inhibition, you're inhibiting the enzyme from working by changing the shape of the active site. And activation, you're activating the enzyme again by changing the shape of the active site to allow the substrates to bind to it again. So you can see where this could be useful in the body. So sometimes we need the enzyme to work, sometimes we don't need the enzyme to work. So you can see when it can turn on and off. All right. And then finally, we have feedback inhibition in metabolic pathways. So at the beginning of our video, we talked about metabolic pathways, which are a series of reactions that take place in order to get an end product. And so sometimes, we have each of these reactions need a different enzyme to occur till we get that end product. And once that end product is reached, we might end up needing some sort of feedback inhibition to prevent the process from taking place over and over again. Okay, so let's say, like the video showed, where we end up with so much of the product that we don't want any more of it. So that end product could actually form an allosteric site on that enzyme to prevent the substrate from attaching to it to begin the metabolic pathway again. And that will prevent the metabolic pathway from continuing. All right. So metabolic pathways, again, are just series of reactions catalyzed by multiple enzymes. But when we have that feedback inhibition, the end product will inhibit the beginning, the upstream process from happening. So it essentially shuts the whole thing down. All right. So that'll prevent too much of that end product from building up. So it shuts down the process. And then, maybe if we run out of substrate or, we need more of the end product, we can have it, release the inhibitor and start again. Okay. So we have these mechanisms to activate the enzyme and so forth. All right, so let's go ahead and do a review question. So which of the following is not an example of an energy transformation? Is it heating up dinner in a microwave? Is it solar panels at work? Is it formation of static electricity? Or is it none of the above? Which of the following is not an example of an energy transformation? Well, aren't they all examples of energy transformation? So when you're heating up dinner in a microwave, so you're using, energy to transfer that, microwaves of energy into heat to make your dinner warm. Okay, so that's an energy transformation. Solar panels, so you're using sunlight, turn that into electricity. Formation of static electricity, so you're using friction to form that little electricity, so none of the above. Here's another question. Which of the following is not true about enzymes? A, they are consumed by reactions they catalyze. B, they're usually made of amino acids. C, they lower the activation energy of chemical reactions, or D, each one is specific to the particular substrate to which it binds. If you answered A, I would agree with you. So they are not consumed by reactions they catalyzed, right? So you have the substrate bind to it, but that doesn't actually use up the enzyme. It just is a little binding site for the reaction to take place. So they are not consumed by the reactions. They can be used over and over again. They are usually made of amino acids. They're protein based. They do lower the activation energy, and they are specific to a particular substrate. So A is the correct answer. All right. So that is the end of 4.3, energy and metabolism. So by now you should be able to explain what metabolic pathways are. You should be able to state the first and second laws of thermodynamics. You should be able to explain the difference between kinetic and potential energy, as well as describe endergonic and exergonic reactions. And you should be able to discuss how enzymes function as molecular catalysts. So this is the end of chapter 4, the working cell. So in lab, we're going to get into some information about how metabolism works. Actually, we're going to get into passive transport. We're going to be looking at diffusion with the potato lab and stuff like that. So we'll get into that particular part of the lab. I haven't done it in a while, so I'll have to look that up. All right, so look for my video on lab number 4, and I'll see you there.